And a few colleges I will mention because they are closely associated to my heart. And when I read some colleges' names and the faculty members, I apologize that if I missed some names. But I was closely associated and I've been here because one of the institute is Sagar, Dr. Sarita Madam. Sarita has been CEO, Dr. Mukesh Dubey, Ankrit, Dr. Shunil. These are all the directors who have been here. RGS, Avinish Tripathi, Nova, Avinesh Varma, and GCRG, Dinesh Chandra, Arya Narendra, DO College, that is Asutosh Mishraji, and Nishant College, Manish, and Dr. Neeraj, and my close, very closely associated, that is Anup Sache from Dayanand College. We have been here, and last 1 week back, he's in Indian Pharmacological Society held in the Pharmacology in New Delhi. And I respected the other members who have been here, sir, because I apologize the names which I have been to be acknowledged here. But when I come into the topic of the present international conference, the conference today is not by the single-handed. It is being associated by the different colleges and the directors, and the core responsibility taken by your director, madam, to come into the topic that is pharma innovation and the advancement in the global scenario. As a pharmacist, we come to know that various disciplines like coming to the diploma, B pharmacy, M pharmacy, and the several disciplines, how we play the key role. As I am one of the pharmacists here, presenting myself as a pharmacist with vast experience of more than 2 decades in this line of research, and I have come from basically from the graduation in the pharmacy from Mysore University, M.Pharmacy from Tamil Nadu MGR Medical University, and PhD from the Banaras Hindu University, and the postdoc from the All India Institute of Medical Sciences. And I have been here from the last 2 decades, more than 20 years, in one of the CSIR laboratory, that is Council of Scientific Industrial Research, that is National Botanical Research Institute. Just I will glance at this is the scenario which we can learn and interpret the theoretical courses to the application oriented, to the industrial oriented, to stand into the global scenario. We have to refine our own subjects of the themes, of the problems that we encountered into the health sector, how we can use our knowledge. Hamara knowledge, what else we are learning, the subjects in theory, and exposed in the different practicals, हम कैसे use कर सकते हैं to the benefit to the human mankind. This is the concept which you have to be taken. This has been rightly directed and driven in this institute now, based on the topic which has been chosen today. In this, I will take you, initially I will introduce what is CSIR. It is one of the premier institutes, that is CSIR. There are more than 37 laboratories in the organizations, in the umbrella of CSIR. When we talk about the state of Uttar Pradesh, there are 4 organizations, and the students of graduation or post-graduation or postdocs, talking about the biological science and the major core area in the application in the pharmacy, will be trained in the organizations into the industrial and application mode of the thing. And we have to use and benefit the things, what the R&D is doing, what the research and development is going on in the laboratory, how to pull this R&D from the laboratory to the industrial oriented and to the public. This is the major clear-cut pathway which we are reading more than 50 subjects in the graduation. And we are reading from the pharmaceutical chemistry, pharmaceutical analysis, pharmacology, pharmacognosy, jurisprudence, law, many things we have been exposed. But when we come into the super specialization and the super specializations, we are parallelly moving to the global concept in the healthcare. Like clinical practice, clinical pharmacy, we are parallelly moving, and we are we are going with the clinical applications and the drug design. And I will, in this concept, I will introduce today antiviral products. This is the global concept today, which is recently, which we are moving, that COVID, dengue, and other things which we are coming across. And what are the natural products? Because now presently I am here, came from the National Botanical Research Organization as a plant-based organization, as a premier organization. Next slide. I will introduce what this organization, how it functions in the CSIR laboratory. Because this gives an idea, not only on the theoretical, you have to come out to see on the global concepts, how the institutes works, and how you can find a space in the organization, and what are the disciplines that you will be taking. And as a pharmacist, you will be reading only pharmacy. When you come into the R&D sector, research and development, and we go for the PG studies, and you go for the PhD studies, and the research and development, there you will see, you will be parallelly moving to the other disciplines, both in the science and also pharmacy and engineering and technology oriented. Therefore, the highly qualified will be competitive to be stand in that platform. There are several disciplines, how it will be monitored, like a research council, management council, what are the sections in the NBRI. Next slide. Next slide. What is the mandate? What are the major R&D areas like plant diversity, systematics, herbarium, plant ecology, environmental sciences? We are having more than 100 acres, that is botanical garden, floriculture, plant microbial interactions, plant molecular biology, pharmacognosy, pharmacology, phytochemistry. But I came in the division of pharmacology, that is pharmacognosy, pharmacology. Next slide. These are the aims and objectives of the organization. Next slide. What are the areas that is specialized services and facilities, like we are having the international and national recognized herbarium specimens of the plants, more than 250,000 plant specimens have been there. Knowledge Resource Center, Central Instrumentation Facility, how do you go for the different spectra and the extractions and isolations of the molecules, and what are the techniques like UV-NMR and many other things of the LCMS and many instruments what have been there, how we can use. Next slide. Now this is the concept of the integrated approach in botanical research. When we look into the subjects, we will be reading different subjects. Because when we talk about the chemistry, we will be talking about only the pharmaceutical chemistry, medicinal chemistry, organic chemistry, inorganic chemistry, analytical chemistry, and we will be correlating the bioassays and the essays in the Indian pharmacopoeia. These are Indian pharmacopoeia, British pharmacopoeia, USP, all things we have been aligned and we are aware. These things, how we attain and maintain the standards to be competitive into the market scenario, both in the national and also in the international world. And these are the things which is like different approaches we will be making, and how do you link up these things to the regulatory aspects like quality control, safety evaluation, efficacy studies, and what are the different techniques and the plants, like for example, when we talk about the plants, there are some plants which are having a huge demand, international demand, and how do you maintain that plants in the scenario of free from heavy metal contamination, and what are the plants, what are the endangered species, what are the plants and rare and endangered species to be protected, and how to cultivate and multiply and supply to the industry, to the economy generation. These are the things which you have to take in. And when we talk about the product development, we'll be talking about the preclinical studies, clinical studies, what are the things that has been taken from the botanical extracts or isolated molecules used for the different therapeutic purposes. These are all things, these are the procedures which is integrated for the different disciplines to come out a drug molecule. Next slide. What are the trust areas of the botanical research? Then, for example, if you talk about the plant from the plant extract, we will be isolating the molecules and reproducing the quantity of the isolated molecule from the different geographical areas, and what is the percentage of that specific marker or the chemical marker reflecting correlately directly proportional to the biological activity. That is the marker analysis, and what has been mentioned in the traditional medicine. This has to be notified. And reverse pharmacognosy or pharmacology, evidence-based research, formulation and development, genomic sequence, systemic biology approach, lead compounds from medicinal plants, pharmacovigilance of natural product, organic farming, plant biotechnology. This is the cycle of the things which is linked to one. Therefore, you have to find the space of your interest and the research to enter into the specific areas to generate your own economy and the space to have their employment. Because this is what is the application oriented which we are reading, it is directly proportional to the employment oriented. This is not the normal, this is the professional course in the pharmacy. Therefore, we have to be, there is a difference between the basic research and applied research. In pharmaceutical analysis, we will be talking about only the pharmacy specialized people will be molded in such a way we will give employment. We are providing employment, and what base we will be providing, how you can become a self-entrepreneur. And if you are specialized in the area of pharmacognosy, you have to talk about the medicinal plants. When you talk about the pharmaceutical chemistry, you will be talking about the standardization and assay. When you talk about the pharmacology, you will be talking about the safety, efficacy of the product. When we talk about the pharmacovigilance, what is the drug-drug interaction, what are the side effects of the drug, what is the monitoring, these all things to be known. When we talk about the clinical practice or the pharmacy practice, we will be talking about the patient care, how to move, how to enter the data, how to monitor the patient. These all things to be codified and recorded and documented, and these all linked to the one of the presentations of the student. I have seen that the artificial intelligence. These are the first areas which is coming up in our scenario in the pharmaceutical line. Next slide. When we talk about the secondary metabolites of plants, there are like plants, like for example, nutrition-oriented plants. Nutrients, like the primary metabolites and secondary metabolites. When we talk about the secondary metabolites, that is phenolic compounds, terpenes, nitrogen-containing compounds. The nitrogen-containing compounds like alkaloids, cyanogenic glycosides, non-protein amino acids. These are the class of the compounds of the plant secondary metabolites which have been well recorded. These things to be standardized and quantified when we are developing the product in this line. Next slide. When we talk about the medicinal plant back to compounds because the presentation is more focusing on the antiviral. Because what are the plants that have been taken, I will be speaking more on the medicinal plants, how will be useful for the dengue and also the SARS-CoV. Along with the marker compounds like butylonic acid, galectin, triglycerin, these are the compounds that have been mentioned in the plant. The effect of the bioactive compounds isolated from plants with the DNV infection. And these are the markers that has been studied, that is like a hemooxygenase, interferons, like non-structural proteins, 5-RNA dependent RNA polymerase, non-structural proteins. Because these are the compounds which are playing a key role to develop the product in the area of the anti-dengue or SARS-CoV. I will be stressing more on how it will be acting, how we will be moving into the drug molecule. Next slide. Like medicinal plants with anti-dengue activity, like Serica papaya leaves nowadays will say that papaya leaves are used, goat milk is used, and how like Tinospora cardifolia is used, guduchi is used, and the neem leaves have been processed. And these are decrease the viral load and viral replication, the effect of the crude plant extract on the DNA infection, how the platelets has been fallen, what is the role of the WBC and the plasma monosaccharides, these are non-structural proteins, TNF-alpha tumor necrosis factor. Because before entering to the other slides, these are the slides of the preliminary traditional knowledge of the Indian context, which the plants has been chosen or been documented in the traditional knowledge, along with the isolated molecules of the specific plant and how it will be used for the dengue. Next slide. Like what? Either the drug has to act as to decrease the viral replication and along with the medicinal plant bioactive compounds with anti-SARS-CoV. These are the plants codified and also documented for the against the COVID. The effect of the bioactive compounds isolated with the plants with active against SARS-CoV infection, angiotensin converting enzyme, ATPase, adenosine triphosphate, 3CA like protease and papain like protease. These are the things which these bioactive molecules acting at the specific sites. Therefore, if you talk about the plant, either you have to choose the multiplication of the plant or isolation of the molecules, and once it is isolated the specific molecules, where it is acting and which protein it is acting. This is the glimpse of the COVID-19 and the medicinal plants which has been codified. Next. When we talk about the dengue, the diagrammatic representation of the possible inhibitory sites of DNV virus, that is dengue. When we talk about the viral genome, that is a core protein and membrane protein and envelope protein. When it is as such the virus, when we infect the host, it converts into the non-structural proteins, that is NS1, NS2, NS3, NS4 and NS5. These are the membrane associated viral replicon, that is serine protease, RNA helicase, RNA triphosphate, RNA dependent RNA polymerase. We will be talking more on the NS3 and NS5. When we talk about the dengue, why we are restricting to the NS and the non-structural proteins and which type of molecules will be acting on the structural proteins. Next slide. This is the genome of the DNA virus and the viral targets. Once virus enters into the human, it converts into the non-structural proteins and structural proteins. That is a cell protein, that is structural protein, that is a non-structural protein. Therefore, we are targeting on the NS3 and NS5. It is more specific towards it. Therefore, the targeted viral proteins, that nucleic acid polymerase, protease, integrase and neuraminidase. Therefore, we will be targeting on the DNA dependent or RNA dependent RNA polymerase and the targeting cellular functions with virus needs. This is the basic. Once you know the structure and the genome of the virus, once the mosquito or the infected once bites on the host, then it will convert it into the structural proteins and non-structural proteins. And when the drugs has to act most on the non-structural proteins, that is NS1, NS2, NS3, NS4 and NS5. And NS3 and NS5 is the more targeted protein. Next slide. Next slide. In the case of the dengue, NS3 and NS5 RNA polymerase are considered the prime target for the antiviral drug therapy development. The 3D structures of the DNA virus NS3 and NS5 proteins. From the data bank we will be taking. These structures were downloaded from the high resolution containing bond inhibitors. And Minstoware is registered as a National Center for the Biotechnology Information that is under evaluation. This is the only drug. To date, no natural compound has been reported against dengue. Next slide. In the initial screening, when we went for the bioactive compounds against dengue, identified more than 135 plant bioactive compounds through literature review. The rationale of selecting the compound is based on the reported antiviral activity against dengue-related diseases, and molecular docking studies were carried out using Stranger software. This is based on this, we will come to know which are the type of the molecules and what are the structure and how it binds to the specific protein, whether it is NS3 or NS5. Next slide. These are once it is binded, then we will come to know the specific molecular compounds and which plant it is mentioned. For example, if it is mentioned that cardifollicide, it is mentioned in the Pinospora cardifolia, which part of the plant that is mentioned in the stem, which type of extract it is mentioned. Whether it is ethanol or a methanol, it is very clearly from this in silico studies, using the software, based on the molecular classification, then it is linked up to the plant, and it is linked up to the which part of the plant and what type of the compound and which type of extract has been processed. Next slide. Next slide. Now, when we talk about the phytocompounds against NS3, when we talk about the NS3 specific proteins, that is phalanthestatin A, what is the docking score and the binding energy, and which type of the plant species it is mentioned. There is herbicitin. These are the plant, that is ellagic acid. These are the reference co-crystallized ligand, is a reference compound. This, based on this co-crystalline ligand, we can say that the high docking studies, what has been mentioned, which type of molecules has been binded to that protein. Therefore, the binding is structural activity relationship. Based on the pharmaceutical chemistry, we studied the CSIR, quantitative structural activity relationship. When once bind to the protein, then what is intrinsic activity and what is the binding energy? These are being scored, and based on this, we can predict that these molecules are having this property of specific protein, whether it is NS3 and NS5. Next slide, please. This is similarly we went to the phytocompounds like NS5 also. These are the phytocompounds source and the docking scores and the binding energy. And based on these in silico studies, next slide. And the molecular dynamic studies has been taken, indicating the stability of the structure during molecular dynamic studies. There is increasing with the amplifier within the first 20 nanoseconds, stabilized for 40 nanoseconds, showing strong stability towards phalanthestatin A and the protein complex. And these then flexible, which ligand binding residues with higher peaks reflects the mobility and active participation in the ligand binding with the minimum fluctuations toward the end of the stimulation. Therefore, from this reference compound, along with the molecule, we will come to know what is the dynamic studies that we are compared with the reference compound. Next slide. Similarly, that RMSD indicates the stability of the structure during when increasing the amplification. Earlier it is NS3 protein, this is NS5 protein. Therefore, from this we will come to know that phalanthestatin A is having a more binding affinity, and the protein complex and the residues suggest that the flexibility in the half-helix backbone. This is the software analysis which has given a clear-cut picture of the molecule which is having, which type of the molecule and how it binds to the protein, and what is the affinity towards it, what is the stability of the molecule while binding. These are been recorded. Next slide, please. In the continuation, this is one of the plant. This is a bioactive compounds. Again, in the bioactive compound, there are different plant molecules. What is their binding energy? What is their inhibition? And what is, when we subject these phytochemical molecules of the plant extract through the NMR, LCMS, and we come to know what are the type of the molecules that are present in the plants. And those plant molecules, when we subjected to the in silico studies, we draw conclusion saying that main protease is a protein binding enzyme for coronavirus that normally breaks the glycoprotein into functional protein for viral replication in the host cells. Therefore, from this we come to know that there are the specific molecules of the natural products acting both on the dengue and also in the COVID, and these in silico studies has given an indication to take up into the in vitro and also the in vivo studies into the pharmacological point of view. Next slide. In this overall, we can see that once the drugs has been identified, and to carry out these, there is a specialized laboratories, that BSL-3 laboratory facility is required to carry out the antiviral studies to monitor the activity of the molecule on the specific cell lines infected either with the dengue or the other viruses. Therefore, these are and followed by the other trials like pharmaceutical. When we say it is a product development, then we see that the preclinical studies and the humans, before subjecting to the humans, we have to carry out on the rats and mice and the preclinical studies. Before that, we will be going for the cell lines followed by the animal studies, followed by the clinical studies. Then it comes into the pharmaceutical drug management, drug discovery, quality control and quality assurance, pharmaceutical manufacturing, and pharmaceutical drug development and regulatory affairs and many other things have been aligned. Uh, Thank you very much for giving an opportunity, and I've been here because this I thought that this is the hot topic which everyone has to be focused on the present scenario to drive our research into the global concept into the antiviral. Once we are thinking, because we are sitting in the hub of the natural Indian scenario, or the backbone is the natural products and the plant wealth of the Indian context. Those plant wealth has to be converted into the product development, into the pharmaceutical application point of view. Once again, thank you, madam, for giving an opportunity.
Thank you, sir, for your knowledge.
I must be thankful to the other faculty members, Neeraj madam, and many. Really great that I have seen most of the people are the faculty here. Thank you once again to everyone. If any questions or anything, once again I have to invite each and every one of the faculty, request from the colleges that students should be exposed to the national laboratories. I request the directors and other faculty members recommend to the CSIR laboratories. You can write a letter from the organization for visiting to the organizations. For example, when we talk about the NBRI, we'll be talking about the garden, we'll be talking about the pilot plant, we'll be talking about the product development. We'll be getting an exposure of more than 100 scientists of different disciplines, from basic level, from the botany, chemistry, organic chemistry, natural products, instrumentation analysis, seeing the sophisticated instruments and be competitive in the global contest of the present theme which has been chosen. In this point, I request all the faculty members to recommend the students to the national laboratories which they get an exposure here. When we talk about the Lucknow, there are more than 5-6 national laboratories, core R&D areas, institutes of the national reputation have been present. One of the institute which I am working is National Botanical Research Institute. Yesterday, madam has said that the other members who has come, like Karna Shankar has come from CMAP, Dr. Karna Shankar, because he is my close friend, and the other members who have been come like CDRI and IITR, CMAP, and Birla Polybotanical Research Institute, and SGPJ, KGMU, because we are sitting in the hub of the national knowledge. Lucknow is the hub of the national knowledge. Therefore, because high intellectuals have been here, therefore we have to see, because when we see the universities also, Integral University, and many other universities which have been projecting many things, that has to be taken the great opportunity to get an exposure both in the educational background with the R&D national reputed organizations. We get a rare opportunity because basically I belong from Andhra. I have not got opportunity to work in national laboratory or to enter into the national labs. But after coming here, I have seen Lucknow is a hub and people are very great. They are getting a good exposure and bringing national scientists or national reputed scientists accessible to the pharmacy graduates. In this, I had a lot of affection in this line. I must be grateful to one and all of the faculty members who are sitting here. This is my request. Thank you once again. Thank you.
Thank you, sir.
If any questions, I can answer.
Anyone has any question?
What you want to become also you can answer, because the topic which I projected is not that you are in the R&D. Our thinking should be high, because it is a global. The theme is more on the global health advancement. Global health advancement means how we stand globally and competitive to be healthcare sector, in the healthcare sector, and what is the need today. How to change our mind setup to enter in the major core areas. Though we study as an antiviral drugs in the 2, 3 or 5 pages or less than 10 pages in our, you do not know your scope afterwards. Therefore, this gives an inclination towards how you can enter into the different areas which you will be choosing in future. Therefore, I focused on the antiviral because little you will get an idea how antiviral studies have been taken, how the drug discovery is moving towards with the antiviral studies, and what like computational biology, like bioinformatics, like artificial intelligence. Like there are several scope of opportunities which you will be working. And the routine things like pharmacology, pharmacognosy, quality analysis, and marker analysis, these are the routine things. But when we look into the concept of the present scenario, how to drive our research towards into the goal of the global concept. This is an inclination I had given that I thought that I have to bring into the minds of the students.
So I'm very thankful that you have given a very interesting and valuable lecture. I'm also a person of pharmacognosy, so my specialization is pharmacognosy and phytochemistry. But today, after listening to you, I see that you are the ocean of knowledge of pharmacognosy and phytochemistry, and simultaneously other fields you are also touching. So my question, two small questions are there regarding antigens, two antigens, NS structural antigens, non-structural antigens. So can you kindly explain what are the difference between the two? And second question is the regarding the in silico study. Generally we are doing the in silico study for single molecules, drug molecules. But can we go for, in case of plants, can we go for the extract in silico study? These are two questions.
Yeah. Sir, it's a wonderful question which you have asked. It's a very broad spectrum. One thing you have to remember, before answering the question, you must understand I am also like you, normal student. Normal student in the sense of not like. When I talk about the research problem, when I touch it, sir has said that you touched several disciplines in the topic. Like pharmacognosy, I touched chemistry, I touched pharmacology, I touched bioinformatics. This is an advantage when we work on when we think and interact with the national and international level of R&D organizations. As my core area is in the area of pharmacology. On my own interest, I've chosen the pharmacology and I've been taking the position of pharmacology as a scientist. But gradually to upgrade to ourselves to the present scenario of the global scenario as per the theme today, it is thought that we have to work on antiviral. Antiviral is a very broad terminology. And specifically speaking, present scenario is on dengue. It is a vector-borne disease. Mosquito. Anti-malarial is different. Antiviral dengue is different. And parallelly, I move to the SARS. Parallelly, we will be moving to the chikungunya. The concept of the things are more focused towards to the antiviral. To work as an antiviral, you must have a biosafety laboratory. But sitting in the lab does not have the biosafety laboratory doesn't mean that we are not working. When we are sitting in the Botanical Institute, we'll be taking the plant and we'll be taking the extract, we'll be isolating the molecule, and what are the plants that has been given in the ancient traditional texts, either in the Ayurveda, Unani, Siddha, whatever the system, it has been mentioned as an antiviral, those plants to be picked up. And what are the chemical markers that are present, chemical molecules that are present in the plant as a secondary metabolites, as I shown in the slide, that it comes under the chemistry of the phytochemistry. When we talk about the plant chosen and nothing, it comes under the pharmacognosy and natural products. When we talk about the specific, the genome of the virus, when I talk about the genome of the virus, it went off into the molecular biology. Therefore, what I done, I recruited one bioinformatics scientist. Dr. Nikesh is sitting here. I am not aware of these things. Because the problem, I know that we have to handle that problem. Then we recruited that specialized core areas of the people. When I talk about the bioinformatics, when I talk about the DNO that software designing and how this protein binding and given that problem to the recruited one student, given the information, and we need this thing to be done. That has been done. And how it is done, like as if it's the virus, the genome when it is entered, when the mosquito bites, the genome will plasma membrane, cell membrane, then it is coming out of the structural proteins and non-structural proteins. When I talk about the structural proteins, non-structural proteins, then when I talk about, given the example of dengue, I said that NS1, NS2, NS3, NS4 and NS5. When I talked about NS2, NS2A, NS2B is there. When I did this point in this conference.